It's good to be back here at CGSI. I know this is the very last day, so we're all very wide awake and alert, which is great. And we introduce ourselves. So my name is Melissa Gymrek. I'm just down at UC San Diego. My lab is really interested in looking at repetitive regions of the genome. And these turn out to be really difficult to look at, which is a big plus for us methods people because it gives us a lot of fun methods things to work on. So we're really interested in how repeats can be involved in complex traits, mostly in humans, but now branching out to other species. And I'm going to talk a little bit about our adventures and doing our first attempts at integrating repetitive regions in a systematic way into GWAS to look at complex traits. So as a field, as human geneticists, we've gotten pretty good at doing genome-wide association studies. If you haven't seen these Manhattan plots before, maybe we're interested in some trait like eye color, and we can collect a bunch of people and look at, record their eye color, and then test each variant across the genome for association with that trait. And for most traits that are reasonably heritable, we can get pretty good results like this, where we can really robustly identify regions of the genome that are associated with different traits. However, our work is not done, and there's still a lot of challenges. I'll point out a couple here. One is that even though we've collected like tens of thousands of these skyscrapers that tell us about these associated regions, for vanishingly few of these do we actually know the full story of like what is the causal variant that is actually driving those signals and making this relationship with the trait. And we're still not really great at predicting most traits in most people. When performing these genome-wide association studies, the standard practice and what we've been mostly doing is focusing on a simple type of genetic variation. You've probably seen these already, which is SNPs, or single nucleotide polymorphisms. So SNPs are really great to analyze because they tend to have only two common alleles in the population. At this example, we have an A and a T. And because there's only two possible alleles, there's only three possible genotypes, and we can easily just encode those as 0, 1s, and 2s, and test for association between, in this case, the number of Ts that you have and the trait that we're interested in. However, common SNPs are really just one way that genomes can vary, and if you take two genomes and compare them to each other, they can actually vary in much more complicated ways. So we've seen SNPs, you can also have indels or insertions and deletions, but there's all this other kind of crazy stuff that can happen. For example, you can have big structural variants that rearrange big chunks of the genome. You can have retrotransposon events. Here we're going to focus on the second type here, which is tandem repeats, or chunks of the genome that are repeated over and over again in tandem, and we'll see that can vary in copy number between different individuals. By the way, this slide is just an oversimplification itself. You can have all kind of other crazy stuff like indels within repeats and repeats within transposons, and et cetera. And these are just some example known pathogenic variants that fall into some of those crazy cases that don't fit into the categories above. So why might we want to look at those other crazy things? Well, it turns out that some of the GWAS signals to date that we do understand, and some of the strongest GWAS signals identified to date, turn out to be driven by some underlying thing that is more complex than just a single nucleotide polymorphism. And I'll just put a couple examples from other groups to motivate this. These are some of my favorite GWAS hits. So one example is in schizophrenia. So this is a Manhattan plot for schizophrenia GWAS. The top hit here is in the MHC region on chromosome 6. But it cannot be really fine mapped down to one individual SNP that is convincingly driving this signal. And in fact, it turns out that this locus is largely driven by these more complicated structural variations in the C4A gene here. And so while GWAS with common SNPs found this region only by looking at this more complicated thing, and here there's 4 different common structural versions of this region, that turns out to be what's the main driver of this particular GWAS signal. Right, and again, common SNPs kind of pointed to the right region, but the true causal variant turned out to be something more complex. I'll just show one more example to motivate this. This is an example from height. So height GWAS, one of the strongest hits for height is over here on chromosome 15, overlapping the aggrecan gene. digging into this signal, within the protein coding region of this gene, there's actually this large variable number tandem repeat, where the repeat unit itself is pretty big. It's 57 base pairs. And if we look at different individuals in the population, number of copies of this VNTR range from about 22 to 29. And if we look at the association between VNTR copy number and height, you see the striking relationship across a range of alleles. And it turns out that the VNTR, the copy number of this repeat here, is a major thing that is driving one of these top GWAS signals for height. In this beautiful paper by Mikhail et al., they go on to show that these VNTRs turn out to drive some of the largest known GWAS signals that have been identified in places like UK Biobank. Okay, so in this talk, we'll be focusing on repeats. I showed some examples of some of these complex regions that can be driving GWAS hits. We'll focus on tandem repeats here. I just want to kind of define what the repeats are that we're looking at and a couple of the reasons why we're excited at looking those on a genome-wide scale. So we're going to look at two different types of repeats. The first is STRs, or short tandem repeats. For STRs, the repeat unit ranges between 1 and 6 base pairs, like the CAG repeat shown here, repeated in tandem. These are found throughout the genome. If you look at the HD38 reference genome, depending on how you count these, you'll find about 1.5 million of these STRs throughout the genome, primarily falling in non-coding regions. We can also think about other types of repeats. So I'll talk a little bit about VNTRs, or variable number tandem repeats. These are repeats where similar to STRs, except for the repeat unit itself can be bigger than 6 base pairs. These also tend to be a little bit messier in terms of having more imperfections in the repeat sequence. These are also found throughout the genome, and again, depending on how you count, there's about 500,000 VNTRs in the HD38 reference genome. So why do we care about these repeats? Well, one reason is that they're found throughout the genome. But another reason is that they tend to have these really rapid mutation rates that make them really polymorphic when we look at them across populations. So I want to look at that a little bit more in the next slide. So we talked about SNPs already back in the very beginning, and we saw that typically SNPs just have 2 common alleles in the population, and that was really convenient. We can just code them as 0, 1, 2 and move on. Tandem repeats are often different in that they often have more than 2 common alleles in the population. Sometimes these things can have 10 or 20 different allele lengths in the population. And so we can no longer code them as 0, 1, or 2, and we need to take into account the fact that there are many, many different possible alleles at many of these repeats. So we combine the fact that these things are all over the genome and they're really highly polymorphic. Tandem repeats are one of the largest sources of genetic variation in humans. But they're technically challenging to look at, and we'll see some of the challenges when we look at some of the applications. Okay, one more reason to motivate why we care about looking at these repeats is that they can be involved in human phenotypes. And some of the most famous examples are in Mendelian disease. And in fact, STRs as drivers of disease were some of the first genetic loci where it was really mapped to what is the causal variant for the disease. Most of these come in the form of what are known as STR expansion disorders. If you're familiar with, for example, Huntington's disease, this is one of the first diseases where the causal gene was mapped. In Huntington's disease, there's a CAG repeat. It's in the coding region of the Huntington gene. Most people have around 20 or so copies of the CAG repeat, but you can sometimes get these large expansions. And if the number of CAGs exceeds 40 copies, then you end up with Huntington's disease, which is this devastating neurological disorder. One thing that's really cool about Huntington's that kind of motivated a lot of the work that we do is that the repeat here is not kind of like a switch, like you have the disease or you don't. You can actually get a spectrum of phenotypes depending on the length of the repeat that you have. So in Huntington's disease, if you look at the number of CAG repeats versus age of onset, well, the larger the expansion you have, the earlier you get the disease, and also the more severe the disease tends to be. So in this case, the repeats kind of act like a tuning knob to control phenotype in a more continuous fashion. Right, so it's well known that these repeats are involved in these Mendelian disorders, but we're really interested in complex traits, like the types of things we would study with GWAS. So these examples kind of motivated us to think about what might be the contribution of tandem repeats across the genome to more complex or polygenic traits in humans. And that's what we'll focus on for most of the talk. So our lab is interested in a lot of different aspects of repeats, and a lot of different kind of things needed to happen in order for us to even look at these in complex traits in the first place. We do a lot of work on methods development for looking for genotyping tandem repeats from sequencing data, either short reads or now moving to long reads. We also look at ways to model mutation and selection dynamics in populations. And finally, we're interested in finding associations between repeat length or repeat sequences and different phenotypes in humans. I will focus primarily on this third aspect here, and I will point out that all of the work that I'm showing is from really amazing graduate students in the lab. Helyaneh and Jonathan are really driving a lot of what is shown here. I'll break this up into two parts. I'll keep track of time. First, I'll talk a little bit about efforts to perform TR analysis in large cohorts, because it's really a prerequisite. If we want to be able to do something like GWAS with these repeats, then we need to have large data sets of genome-wide genotypes of these repeats across population or biobank scale. And then I'll talk about our efforts to integrate STRs into GWAS and some of what I think are pretty exciting associations and signals that we've found with that. When I was a graduate student and I proposed looking at tandem repeats from sequencing data, my advisors on my thesis committee kind of laughed at it and thought it was crazy and too hard to do. But really over the last 10 years or so, there's been kind of an explosion of methods that now enable us to get pretty robust tandem repeat genotypes from sequencing data, even from short Illumina reads that might not actually span across the entire repeat region that we're interested in. So I put a couple of examples of these tools up here. All of these do some kind of flavor of taking in the sequencing reads of an individual and giving out a VCF or a VCF-like file that has the estimated diploid copy number at each TR across the genome. And so really the starting point for a lot of our analyses is a big matrix of genotypes where for each STR and each individual in our cohort, we get the copy number and in some cases the sequence of the alleles that at the different repeats in our reference genome TR set. So I mentioned in the previous slide that there are quite a few tools now for doing this genome-wide TR analysis. But what we have found is that really there's not a huge amount of overlap between these different tools that are out there. And so I just want to point out kind of some of the challenges that we face in doing this, is that really just defining what constitutes a TR in the first place is really challenging. So for example, some of those tools on that previous slide just look at STRs. Some of them look at some STRs and some VNTRs. If you look across different tools and how they just define what are the boundaries of a TR, even that can be challenging. So here's an example where a repeat, this is a tetranucleotide repeat, but there's some imperfections in here, and different tools will define the boundaries of where that repeat falls differently. Here's another example. Some tools will only consider perfect repeats, but other tools like Hipster will take into account the sequence imperfections that are kind of this kind of looks like a repeat, but it's really kind of messy. And we're really appreciating more and more that a lot of repeats in the genome actually look like this messy case here. All right, so if we look at the overlap between the repeats that are considered by these different tools that are out there, the overlap is actually kind of strikingly low. And in fact, we looked at the reference sets that are genotyped by these different tools that are out there. And the majority of repeats are actually only considered in the reference set of one of these different tools. So if you run only one method for looking at repeats, you end up actually missing a lot because there might be hundreds of thousands of repeats that another tool would genotype that just weren't considered to be repeat enough to be in that tool's reference set. So our latest effort has been, if one tool's not good enough, then we try all of them. And so our latest efforts have been using an ensemble-based approach. So we have a new method called Ensemble TR, where rather than just relying on any individual one of these methods, which each one is really missing quite a lot, we instead can call all of these methods, and then we have a way to integrate the call sets from each of these different methods into a consensus genotypes. And I won't go through the methods of this Ensemble TR method. This is up on bioRxiv, but we attempt to go through the output of each of these tools and find loci that were called by multiple tools and merge them together. And we have a voting-based method that takes into account the quality scores and other metrics reported by each tool to try and get a best-case genotype that ends up performing better than any individual single tool. I won't talk more about the Ensemble TR method because I want to get to the complex traits, but I will just give some characteristics of the call sets that we can get with this. So we end up getting around 1.7 million TRs when we combine these different individual tools. We've recently run this on around 3,500 samples from the 1,000 genomes data set and the H3 Africa cohort. We've done a lot of work to do PCR validation on more than 1,000 individual calls at these loci, and all of the data from this call set is available at our site called webster.ucsd.edu. Okay, a couple more things before we get to complex traits. One thing, I just want to show an example application that we can use now that we have this big data set of TR genotypes. One example application that we used it for was looking for population-specific expansions of these repeats. Let's look at one example here. So there's a CTT repeat that's upstream of this gene, an EXN. We can use our Ensemble TR calls to look at the distribution of copy numbers of the repeats across different populations in 1,000 genomes in H3 Africa. And we found this and several other examples that we see like this long tail of expansion alleles that are only found in certain populations. So in this case, there's this long tail of like really huge alleles at this repeat that are only found in the African populations in our data set. And one more cool thing that we found at this locus, but actually many others that we've looked at, is that until now we've really been focused on repeat copy number, but we really need to be looking at repeat sequence as well. Because when we look at these huge expansion alleles, and we go over and look at PacBio hi-fi data for some of these regions, if we manually inspect the reads, we found that actually, even though this is annotated as a CTT repeat in the reference genome, there's actually many of the expanded alleles here have this totally different repeat sequence, so CTT, CTC, so it's a hexamer repeat. So not only did the length explode, but in many of these repeat expansions, the motif, the repeat unit itself, is actually changing itself. And that also turns out to be important for some of the new repeat expansion disorders that are coming out as well. So really motivating to take a look also at sequence of repeats in addition to the length. Okay, one more application before we get to the complex traits. So we really, I motivated this by looking at GWAS. We really want to be able to look for associations between these repeats and different complex traits. However, for most GWAS data sets that are available, we actually don't have sequencing data. What we have is SNP genotype array data. This is changing with whole genome sequencing data sets for UK Biobank, for example, but I'll point out some challenges with that in a bit. So in most cases, we actually just have the SNP data. But now that we have these genome-wide panels of TR variation and things like 1,000 genomes, we can actually use that to phase the STRs onto SNP haplotypes. Right? So we can know, okay, this combination of SNP alleles is correlated with having this length of repeat allele here in the middle. And we can use that to impute repeats into SNP data sets where we couldn't directly genotype them. And even though we thought that this might not work that well, it turned out for the majority of repeats, this actually works pretty well. This plot is looking at the number of common alleles at each repeat versus the concordance between the imputed and the genotyped calls from Ensemble TR. For most repeats that have just a couple of common alleles, which is actually the majority of them, the concordance is around 99% between the imputed and the genotyped calls. Of course, this gets much harder for the craziest repeats. So for example, the Huntington's locus can have, something like 20 different alleles that we are looking at. For that one, the concordance is lower. It's around 80%. But still, most of these we can impute pretty well, and usually pretty well enough to go and use them as input to genome-wide association studies. Okay. So now we have these methods that we can genotype these TRs on a genome-wide scale. We can also impute them to data sets where we only had, say, SNP array data available. Now we're ready to go and try and integrate these things into genome-wide association studies. Okay, so we're going to attempt to integrate STRs into GWAS. How is STR-based GWAS going to differ from SNP-based GWAS? Well, in SNP-based GWAS, we've already seen an example of this. Usually there's just 2 common alleles in the population. We code that as 0, 1, and 2. And we test for association between those 0s, 1s, and 2s and some trait of interest. For STRs, we're going to do like the simplest thing that you could imagine, which is very similar to the SNP case. But here, the only difference is that instead of 0s, 1s, and 2s, we're just going to simply take the length of the repeats of the 2 alleles that each person has and take the average of those. So here we're really assuming that everything is kind of additive and linear in terms of copy number association with trait. I'll show a nonlinear example at the end for where this doesn't really work out. But for the most part, we're looking for these linear associations where we can simply do a linear regression to test for association between copy number of the repeat and some trait of interest. Okay, so Jonathan Margoliash in our lab has really done a heroic effort to try and do like the first STR-based GWAS in UK Biobank. So for this, we took around 400,000 white British UKB participants. We used our reference panel to impute around 500,000 STRs into the UK Biobank. And then we tested each of those STRs for association with a bunch of these different blood and serum biomarker traits. In parallel, for comparison, we did the same thing with the imputed SNPs and indels that were available from UK Biobank. Right, here's an example of the results that we get out from doing this STR-based GWAS. So these are Manhattan plots for 2 example traits. And these Manhattan plots, blue dots are the SNPs and indels. The orange dots are the STRs. And for the most part, we can find that the signals that are being pointed out by the STRs tend to overlap with the signals that are identified by the SNPs. This makes a lot of sense because we actually use the SNPs to impute the STRs, so everything is kind of correlated with each other and identifying similar regions of the genome. So we're not really identifying like brand new genome-wide significant signals. In some of these cases, we found that the STR might have a much stronger p-value than any of the other variants nearby. So this motivated us to dig into some of these individual signals and try and understand example signals that might actually be causally driven by an STR. I do want to show you an example of what one of these looks like. So here's an example TR trait association that pops up. So this is a CGG repeat that's in the promoter of this gene CBL. And we see this really nice correlation between the sum of the allele lengths, so the length of the CGG repeat, and in this case, the mean platelet count across the population. I'll note that these are kind of like really fun to look at because we get, instead of just like 0, 1, and 2 with 3 different categories, we get a whole allelic series, like a whole range of allele lengths to look at. And so when we see this kind of monotonic trend with repeat copy number versus the trait, it's really kind of exciting for us to see. Okay, so we've done, we found these signals by doing this STR-based GWAS. And I mentioned that if we zoom in on these individual signals, in most cases you would find like both a bunch of SNPs and a bunch of STRs that would be associated with the trait. And so actually what we spent the most of our time trying to figure out is like which of these signals is actually causally driven by an STR. Because everything is correlated here, it's kind of hard to look at any individual one and say that one of these is actually the driver of the signal and it's not just tagging something else. And so I'm running out of time, so I won't say a whole lot about like our fine mapping pipeline, but I will say that we spent a lot of effort to try and fine map these individual regions to identify what the causal variant might be. We found that especially since STRs tend to be kind of difficult to impute, so the genotypes are a little bit noisier, that running different fine mappers or even just like changing the parameters of individual fine mappers often could give us like very different causal probabilities for the individual variants in our data set. And so I'm not going to go into details for the pipeline that we used, but we basically ran 2 different fine mappers and we poked every parameter we could determine that is pokeable. and only decided to take STRs that passed all of those different settings that we tried with a reasonably high posterior probability of greater than 80%. All right, so after doing all that, we ended up with 118 STR trait associations. I'll show some examples, but these include some of the top GWAS signals for some of the traits that we looked at. For example, a protein coding CAG repeat was one of the top signals that comes out for LDL cholesterol. And altogether, we estimate that STRs explain about 5 to 10% of the GWAS signals that we identified for these traits. And I'll spend the remaining time, I just want to show a couple of examples here because they're pretty fun to look at. So I already mentioned the CGG repeat in CBL associated with platelet traits. I want to show a couple more details about this locus because I think it's pretty cool. So one thing that we found in this locus, and again, this is kind of a recurring theme, is that in addition to the repeat length, there's also a sequence difference. So this is actually annotated as a SNP difference, where some people have this TGG interruption in their CGG tract. For most of these that we've looked at, the distribution of alleles can turn out to be kind of really population specific. So this is kind of hard to see, but for instance, Black individuals from UK Biobank tend to have much shorter alleles at this locus. And this imperfection, which is marked by the hash symbols here, tend to be more prevalent in the longer alleles in the population. If we look at this signal and we instead break it up and look separately at individuals that did or did not have that sequence imperfection and the repeat, we see that there's kind of like two independent things going on here, where if you had that imperfection in your repeat, you actually have elevated platelet count compared to people that didn't have that imperfection. And after doing additional fine mapping of this locus, the combination of the repeat length here and the sequence imperfection could completely explain this particular signal for platelet count. I'll show a couple more examples. I'm keeping track of time. Okay, here's another exciting example. This is a homopolymer repeat. Homopolymers, so this is like a bunch of A's in a row, are some of the most challenging regions of the genome to look at, no matter what technology you're using. But this is a case where we found, this is an intronic poly A repeat in this gene TAUC1, where we found this really striking association between the copy number of the homopolymer and in this case mean platelet value. Here's the original signal in UK Biobank, this Manhattan plot. The p-values here are really crazy. They go to 10 to the minus 300, and that's kind of where it caps out beyond the point where we could get precise p-values. If we just condition on the sum of the poly A tracks that each person has and regress that out, that completely kills the signal at this locus. So here's an example of a really strong GWAS signal for this trait that was completely explained by the poly A length at this region. I don't have time to talk about more cool examples, so I'll just flash forward to the conclusion and challenges. I did want to talk about a couple of challenges that we faced. One challenge is that we've really only been focusing on linear associations between repeats and traits. Turns out there's some really cool nonlinear examples that we found. Here's one for HDL cholesterol. This is what happened when we summed the total repeat lengths and tested for association. We got this crazy trait. If we break up the two alleles from the two different haplotypes and look at those separately, it looks like this is really driven by this nonlinear association between copy number and the trait. This is one cool example. And I'll just end by talking about some challenges that we still face. So, genotyping TRs, right now we were relying on imputation. We'd really like to move to whole genomes. Doing that on the cloud for UK Biobank from 500,000 whole genome sequencing data sets would cost literally $200,000. And so that is one area that we really need to improve, is speeding up these tools if we want to really be able to do that at biobank scale. I mentioned that our current pipelines only deal with linear associations between length and a trait. We ignore, for the most part, nonlinear things or sequence imperfections. And finally, the fine mapping is still a real challenge. If everything is all linear, then everything kind of works out. But if we deviate from that, then the fine mapping assumptions kind of break down, and I think there's more methods development to do there. I'm running out of time because we started late, so I will just conclude. I have time? I thought it was till 10. Oh, it's till 10? Oh, I was thinking it was till 9:45. Oh, okay. So I can stall. I can tell a joke. Okay, well, I'll show you like one more example, and then we have time for questions. I don't know why I got confused about the timing. Okay. Okay, here's one more example that we found. This is a polystyrene repeat in this transcription factor E2F4. So this is one of the protein coding ones that we found. This polystyrene repeat turned out to be associated with 4 different traits in UK Biobank. And each of these, there was this, again, kind of like beautiful association between copy number of the repeat and each of these different kind of 4 blood traits. We're really kind of interested, and now beyond just like finding those associations, what they might actually be doing. And so for the protein coding ones, this is kind of like all speculative, but we have been playing around to see what we can find out with AlphaFold from looking at some of these repeats. So you know, we're still kind of experimenting with this, and these are just hypotheses, but I'll show you one example of a hypothesis that we've gotten from playing around with AlphaFold on some of these repeats. Okay, so in AlphaFold, you can enter the sequence of different alleles of the protein that you're interested in. So in this case, we took that E2F4 sequence with varying number of polystyrene repeats here. And then you can overlay the structure on top of each other and see if the structure is changing at all with the AlphaFold. So here we did just one very short allele and one very long allele. And those didn't, and here the red region is showing where the polystyrene is here. And that didn't really seem to change anything with the protein structure here. But then after doing some more literature search on this particular repeat, or on this particular protein, we found that it tends to have a cofactor that's usually bound to it, this RBL2 protein. So then we put into AlphaFold not just this E2F4 sequence, but also you know, the combination of these 2 sequences to see if, well, maybe differences in the repeat length would be affecting how well these 2 things bind to each other. And so these are kind of hard to see, but the top is looking at the short allele, and the bottom is looking at the long allele. And what we found, and you have to rotate these kind of like to see it well, is that for the longer allele, actually these 2 are bound together much less tightly. So we have this hypothesis that the long poly S allele destabilizes you know, this protein by making it bind less well to this RBL2 protein that protects it from being degraded. So now we have a hypothesis that we can go and, you know, test in the lab or test with proteomics data that, you know, maybe this polystyrene is affecting you know, how much of this protein gets degraded and ultimately affecting its protein levels in the cell. Okay, so I went through a couple examples. We saw the CBL example, we saw the TALC1 example, we just saw the E2F4 example. We have, you know, kind of a big handful of these examples to dig through. Many of these show, and these are just a couple more of these because I think they're really beautiful to look at. Again, many of these show this kind of beautiful allelic series between copy number of the repeat and the trait that we're interested in looking at. Again, these have all focused on just looking at, you know, these linear associations. We have a lot more work to do to like dig into these sequence imperfections or these kind of nonlinear trends that might be driving some of these signals. Okay, so I can maybe spend a little bit more time on the challenges because I'm no longer running out of time. So I mentioned that, you know, our genotyping here focused on the imputed genotypes. But now there's actually whole genome sequencing available for the UK Biobank where we could actually just go and directly genotype these things. And in fact, we've actually done that for a subset of the repeats that we found to be associated with traits here. The imputation quality looks quite nice. But even just running on the cloud for UK Biobank for 600 or so loci to validate the imputation costs like many thousands of dollars for us to set up our pipeline in this cloud and figure out how to run things on DNA Nexus. So really looking forward, we're thinking about ways to speed up these methods for doing genome-wide TR analysis. I already mentioned how we want to look more at these nonlinear things and these sequence interruptions and how fan mapping is kind of really challenging. One more thing I will note is that we do make a big effort to make this not only to perform these analyses, but to make tools that other people can go and run these same things themselves. I mentioned some of our genotyping methods like Ensemble TR, but one of the toolkits that I'm most proud of is this TR tools Python package that we have up, where we're kind of putting all of our new methods that we develop are being integrated into this package. These are tools that support VCF output not only from our own TR genotyping tools, but from other popular genotyping methods like Expansion Hunter, for example. And we have a whole range of utilities that can kind of standardize a lot of these analyses. So for example, if you want to do merging of STR VCF files across samples, or if you want to do filtering to have more standardized QC, we have Dumpster for that. The most recent, the GWAS efforts use a tool that we put here called Associator. So if you have your own STR genotypes and you want to do association tests, these tools are available to do that, kind of moving towards a standardized way to do some of these analyses. Okay, so I will now just conclude. Really, I think both with the short read data, but now especially with the advent of long read data that can span across even the most complicated repeats that were just impossible to look at with short reads, we're really entering a golden age of discovery in terms of looking at these more complex regions of the genome. And I think it's really exciting to see all the example signals that have been pointed out, not just by our own work, but by other work, especially in the VNTR field, where some of the strongest GWAS hits that we know about, again, are often driven by these more complicated things that we didn't initially think to look at. I've mentioned a couple of future directions, but really we focused here on getting this pipeline set up and running it on these blood traits, which are some of the easiest traits to look at that have the strongest signals, that are the cleanest data. But we really want to expand and go beyond the blood traits that we've looked at to look at more medically relevant traits, to expand this to more diverse populations. And then, of course, to do follow-up of some of these hits that we've identified to figure out what is the mechanism by which this repeat copy number or repeat sequence is actually driving changes in these traits. And finally, I mentioned with the TR tools and other packages, a big focus of our work has been making tools and data sets that are available to the community. So we have this TR tools package. I also mentioned our Webster site, where you can go and kind of browse different population-specific allele frequency and other type of information. Please reach out to me if you're interested in using any of those, and I'm happy to help. Finally, I just acknowledge all the wonderful people that worked on these projects, especially Helyaneh and Jonathan, who drove a lot of the work that was shown here. And now there's actually time for questions. So.